Membrane fluidity. The membrane of cells is not a fixed, firm thing. It is flexible. It is fluidity. It has fluidity. And these glycolipids and phospholipids in them, they can rotate and even wander to the other side of a cell within just a few minutes. And there's all the time movement. And depending on how much fluidity there is on how much double bonds are in the double fatty acid tails of the phospholipids and glycolipids, and also how much cholesterol is there. And they seldomly flip-flop to the other side, but they stay there like that on the one side of the lipid bilayer, with a non-polar region in the middle. That is why they don't flip over. These glycolipids and phospholipids, they have all, the phospholipids have phosphor head, which is polar and hydrophilic, and the glycolipids have a carbon head, which is also hydrophilic and polar, and they all just want to be there to the outside, to the extracellular fluid, the watery part of the outside of the cell, and the other ones on the other side of the lipid bilayer want to go to the cytosol, the fluid part on the inside. In the middle, that is hydrophobic, they don't want water, non-polar, so they're not changing the sides. All these glycolipids and phospholipids. Glycolipids are anyway just on the outside. And they are moving to the side, so it is able to puncture such a cell, they're going to the side and going in again. And that makes it possible to make fertilization, to go in with something and change it, put something in and go out again, or also cloning. Dolly the sheep was able, because you can go in with a needle and exchange the thing in it, to put another one in, and then you can clone. So the things are moving to the side, they're moving and closing in again. And the cholesterol is it which makes the fluidity down. If it's a lot, the cholesterol makes it more firm, but also makes the fluidity down. And by cold-blooded animals, that has an advantage because it works the other way around at low body temperatures. It's not explained how and what, but it could be like that. Cholesterol in a warm-blooded animal like humans, where it makes it more firm and less fluidity. In cold-blooded animals, reptiles, lizards, whatever, snakes, where it makes it more fluid at low body temperatures, as snakes can have. And then if they are in cold mode, they still can have fluid blood, because the cholesterol makes it there at low temperatures more fluid. Not in fixed, not in higher temperatures, where it makes it more firm, less fluidity in humans. And that is why one idea why cholesterol can be a problem, too much of their excessive, that it might cause atherosclerosis. The cholesterol is bonding with its OH polar head to the polar heads of the phospholipids and glycolipids, and makes it more stiff, less fluidity. And if there's too much of them, it is too less fluidity there, and the cells lining the blood vessels are having not so much fluidity. It's hardening of the arteries or of vessels, atherosclerosis, and that is the idea that too much cholesterol can lead to atherosclerosis. And that is according to Gerard Torres and Senegabowski's Principles of Anatomy and Physiology book on the pages 63 and 64, but this is no substitute for medical knowledge, and also no medical advice. Hi and welcome. I'm trying to learn biochemistry today. Membrane fluidity, and after we've done with it, we're gonna do arm strength training. And if you participate in the arm strength training, then it's better to always. Membrane fluidity. Membranes are fluid structures, rather like cooking oil, because most of the membrane lipids and many of the membrane proteins easily rotate and move sideways in their own half of the bilayer. So the first thing, they are fluid structures. I never heard someone even indicating that they are fluid structures. Now I'm questioning, do we actually have to take them by the word fluid structures? We're seeing fluid structures. A bit like cooking oil, we say. Many of the membrane proteins easily rotate and move sideways in their own half of the bilayer. Neighboring lipid molecules exchange places about 10 million times per second and may wander completely around the cell in a few minutes. So the thing is really not like a fixed thing. It is more like the tortoise, which we did in Roman Empire studies. It is basically just a group of people standing out with their shields, and if they want, they can just switch and another person is standing. That is interesting. So, neighboring lipid molecules, these lipid molecules, which are all standing there, they are exchanging places about 10 million times per second, but I don't think a single one. Altogether 10 million times? but it wouldn't be enough. Whatever they know exactly mean with it, they are able to wander once around the cell in 1 minute. So basically, we can run around the cell in 1 minute. Membrane fluidity depends on both the number of double bonds in the fatty acetates of the lipids that make up the bilayer and the amount of cholesterol present. Cholesterol, that was sometimes, to explain it, about 75% of membrane lipids are phospholipids, lipids that contain phosphate groups present in small amounts of cholesterol, a steroid with an attached alcohol group. To explain what the cholesterol is doing. The remaining 20% of plasma membrane lipids are weakly amphipathic. So one part is hydrophilic, the other part hydrophobic. Are weakly antiphatic cholesterol molecules, which are interspersed among the other lipids in both layers of the membrane. The tiny OA group, yes, the cholesterol has an OA group attached, is the only polar region of a cholesterol. So that region is attracted to water. I think the other one is want to go away from water. And is formed by hydrogen bonds with the polar heads of phospholipids and glycolipids. The stiff steroid ring and hydrocarbon tail of cholesterol are non-polar. They fit among the fatty acid tails of the phospholipids. Also, the other way around, the non-polar part wants to go to the water. So they're sticking between all these other phospholipids. And these phospholipids, they're on both sides. And the head of them wants to go to the water, the tail wants to go away from water. And the other one also. And they're basically, they are, it's that simple, I didn't realize it. There are just 1 million of them. Let's say 1 million. There are just 1 million of them. And now someone is coming and putting water there, and they're all turning around like magnet. And they're all turning around, facing with the tail to, with the head to the water. But it happens that on the inside is also water, so the other half is turning around. And they seem to be able to switch 10 million times per second once around the cell. So we're just lying around there. There's a magnet, there's some attraction of water at the one side, and attraction at the other water, and we're just lying there, and suddenly we're turning around. One half faces with the head to the inside of the cell where the water is, the other one with the head to the outside where also water is. That is it. They are not sorted there, they are not fixed there, they are just there. And there is just a force which pulls at them, like a needle of a magnet, which is also turning always to the north, or other things, just a bunch of needles which are magnetic, just a magnet here and here. One half is turning around, the other half is there turning around. Why the half? Because we're fitting just a place out. They are just there. But that is all. These are just a big needle, stick of needle. What is we saying? Finding a thing in the needle in the straw stick, or what it is. It's just a bunch of needle stick, and they're all just turning around with their head towards the water part. Because I think it was because the one has hydrogen, the other one had oxygen. The water has oxygen in it, and the head of this glucosylphid is hydrogen. The hydrogen wants to couple up with the oxygen because then they can form bonds. It was some explanation like that. It's the explanation why we want to turn to water. Okay, membrane fluidity. Membrane fluidity depends on both the number of double bonds in the fatty acid tails of the lipids that make up the bilayer of the amount of cholesterol present. So how much cholesterol is bound to them has an influence on how fast they are turning. Each double bond puts a kink. They used that word somewhere else before. I think it was in medicine. I don't know what it was mean. Each double bond puts a kink, in quotation mark kink, in the fatty acid tail, which prevents lipid molecules from packing tightly in the membrane. I understand it in the way that there is something broken. There is a shattered thing. There's something broken. And now we can't turn which much. Because of the fluidity of membrane lipids, the lipid bilayer self-seals it. The lipid bilayer self-seals if it is torn or punctured. Yes, it just goes back again together. When a needle is pushed through a plasma membrane and pulled out, the punctured side seals spontaneously and the cell does not burst. These properties of membranes allow scientists to fertilize an ovum by injecting a sperm cell through a tiny syringe or to replace a cell's nucleus in a cloning experiment such as was done to create a cloned sheep named Dolly. Or they're saying it, this thing just immediately closes again. I don't really know. There are many examples, so I don't really have to say, but it's just, it goes to the side, it goes in, and it just closes again. There are these, I don't know all the names of them. Like the thing that people have hanging in a door. You put it to the side and it just folds again closed too. Despite the great mobility of membrane lipids and proteins in the other half of a bilayer, they seldom flip-flop from one side of the bilayer to the other. That makes sense. If it wants so strong hanging with the head to the hydrophilic side, you can punch, you can, whatever, you can push something through it. They might go to the side, but they're not turning around. Why should they? It's way more complicated. They say the flip-flop from one side, from one half of a bilayer to the other. This is because the hydrophilic parts pass through the hydrophobic core of the membrane only with great difficulty. Yes, the center is hydrophobic because there are all the tails of these glycoproteins, and the head doesn't want to go there. The head is hydrophilic. Why should the hydrophilic part turn in the center between these two lollipops or whatever? They don't want to go in the middle. Because flip-flopping is rare, the halves of the membrane bilayer remain asymmetric. So they're asymmetric because they don't flip-flop. This external half contains some lipids and proteins that are structurally and functionally different from those in the internal half. It makes sense. It is in contact with the outside. So some things are different. Like a wall. It's just whatever. It doesn't make sense, my example. The things in there have access to other things. So the chemical elements sometimes would do other things with the outside world. Because of the way it forms hydrogen bonds with neighboring phospholipids, the membrane on the outside, because of the way it forms hydrogen bonds with neighboring phospholipids and glycolipid heads, and fills the space between and fatty acid tails. Cholesterol makes the lipid bilayer stronger but less fluid of normal body temperature. Because flip-flopping is rare, the halves of the membrane bilayer remain asymmetric. The external half contains some lipids and proteins that are structurally and functionally different from those in the internal half. Because of the way the external half of a bilayer forms hydrogen bonds with neighboring phospholipids and glycolipid heads and fills the space between bent fatty acid tails, cholesterol makes the lipid bilayer stronger but less fluid at normal body temperature. So the cholesterol at the outside makes it more stronger but less fluid. Okay, let's just see that. Interestingly, at low body temperatures, cholesterol has the opposite effect. It increases membrane fluidity, which is important in cold-blooded animals, but not in warm-blooded humans. We're talking of human beings. Plasma membranes accumulate an excessive amount of cholesterol, they become stiffer and less flexible. One consequence of atherosclerosis, commonly called hardening of the arteries, is accumulation of cholesterol in the plasma membrane of cells that line blood vessels. The added cholesterol stiffens the plasma membranes, thus contributing to the decreased flexibility of blood vessels that characterize this disease. Right over blood vessels. The added cholesterol stiffens the plasma membrane, thus contributes to the decreased flexibility. Why of blood vessels? Why not just of blood cells? Okay, whatever. It makes it. We understand what's going on. Now, I want to say at that point, because we're going a little bit into medical things, the books from the early 2000s, it could be that there's more known. But for the same year, I think the basic science is still the same. Cholesterol is going to be outside the membrane of a bilayer. I think of that bilayer sticking in there because it sticks in there. It's not so much fluidity, and a basically hardener. And that they say, I don't know if that is no better known, but they say that has an influence on making the blood flow not so good, or hardening also the artery walls. I don't know why no, not just the blood cells. Whatever, that hardens basically that the outside cell is not so fluidity because of the sticking cholesterol in it, and that can have a part in atherosclerosis. Now, that was the biochemistry part. If you're here for biochemistry, that was non-medical advice, and I want to say here, that information, I don't know, it could be that there's more known to cholesterol in the blood and so on, but the basic principles, it is interesting. If you stay here and participate, that was the biochemistry part. If you go to the next one, we're going to talk about biochemistry. If you stay here and participate in the Armstrength training, this will be honest. The next exercise is shoulder forward apart with and release the fingertips as far to the sides as you can. And down, reach again. And again, reach in front. High up. Again down. And fists and circle fists. If this exercise causes pain, then don't do it. And back again. And now the other way around. And now the first exercise. Press the fingertips together. You don't have to do this with full force. You shouldn't. If this have a little bend in the elbow, if this causes pain anywhere at the elbows, the wrists, the fingers, or in particular the finger joints, then stop immediately. And you don't have to use full force. The muscles of your arms are stronger than the joints of your fingers. And we don't have that much time with some of the fingertips. And the next exercise, press with the fingertips into the fingernails of the other hand. If this causes damage at the skin of the hand or at the nails of the fingers, then don't do it. Press and give. And give. Press and give. If this causes any other pain at the wrist or something, then don't do it. Press. And now we are very around. With the left hand forward up. Press and give in. Press. And now one hand out, press, and give it again, not full force. Press, in, press, in, and again press, and in. And now the square, press with hands together. If this causes discomfort or pain at the wrists downward, press, not full force, go over, up, over, down, middle. And again, back, over, up, over, down, back. Now the rhomboid, middle, up, over, down, and back, over, up, middle. And whatever that is called. Arms to the side, circle them in front, and also backwards. And one arm in front. Shoulders back and down. Pull the arm and have flat fingers. Relax the arm. Go it back with the shoulder, pull at the arm, turn the power to the sky. Other arm in front. Shoulder back and down. Pull the arm and have flat fingers. Relax the arm. Go it back with the shoulder, pull the arm and turn the palm up to the sky. And go with moderate pressure over the middle part of the lower arm. And on the other side. And that was the Armstrength training. I thank you very much. And until next time.